So, my name is Manuel Viotti. I'm at the Zouves Fertility Center. And today I'm going to be talking about, thank you, about this new category in PGT-A, which is mosaicism, really. At the Zouves Fertility Center, we've been involved with PGT-A, or when it was called PGS still, using fish, and then we transitioned to arrayed CGH, and now we use next-gen sequencing, which should be the only technology really to diagnose mosaicism. And the platform that we use is the Illumina VeriSeq or Vitrolite VeriSeq. So all the data I'm going to show you today is generated with Illumina VeriSeq. So I'm going to be talking about mosaic embryos, and what I mean is blastocyst stage embryos that have a mix of chromosomally normal and abnormal cells. So as you know, with PGT-A, we collect a few cells of the trophectoderm. Let me see if there's a pointer here. It doesn't work on the screen. We collect a few cells of the trophectoderm and analyze those cells, and they serve really as a proxy for the remaining embryo. Those are two profiles. On the left, you have a profile of a euploid trophectoderm biopsy, where you see the values for all the chromosomes labeled on the x-axis. They hover around copy number 2 until you reach the Y chromosome, which drops to 0 because this happens to be a female. On the middle, we have a aneuploid where for chromosome 16, it's a monosomy, so the values for chromosome 16 hover around copy number 1, and that's that red line. And also you have copy number 1 for chromosomes X and Y, because this is a male. But sometimes we get this type of profiles, and this is consistent with mosaicism, because if you have values that are between whole chromosome numbers, for example, for chromosome 2 here, the values are between whole numbers. So there's a range of 20 to 80 that has been kind of recommended by PGDIS, because this is consistent with what you would expect with mosaicism in a trophectoderm biopsy. So this would mean that there's some cells in the trophectoderm biopsy that are normal, and some are abnormal, and they happen to be monosomic for chromosome 2. So question is, what do we do with these embryos? And I just showed you an example of a intra-biopsy mosaicism. So we can actually pick up the mosaicism within the biopsy that we collect. But in PGT-A, we can also face this other scenario, which is a global mosaicism. So the biopsy looks uniformly aneuploid, for example, but the rest of the embryo is euploid. So this would be a false result. And how do we resolve these? And how often does this happen? It's a true worry, really, for PGT-A. So let's discuss this scenario first. So how representative is a trophectoderm biopsy for the remaining embryo, specifically the ICM, which will give rise to the fetus and then to the baby? So in order to get to that question, we rebiopsied 93 blastocysts that originally were uniformly aneuploid from the trophectoderm biopsy, and then rebiopsied them in the ICM and then compared the results. So this is a summary of the results. The navy blue dot, so each dot represents one embryo, and the navy blue ones means that the trophectoderm biopsy was perfectly concordant with the ICM. The light blue ones are the status of ploidy is exactly the same, so both were aneuploid, but the aneuploidies could be slightly discordant, so there was an extra segmental or a mosaic region. There were two embryos in beige where the trophectoderm biopsy was aneuploid and the ICM was mosaic, and one sample where the ICM was actually euploid. So from this we conclude that 90 out of 93 embryos are concordant in terms of ploidy. So this tells us that a PGT-A is highly predictive when you use a trophectoderm biopsy. And this was published earlier this year. So let's entertain the second scenario where you pick up the actual mosaicism in the trophectoderm biopsy. So how good is PGT-A at really detecting mosaicism, a mix of normal and abnormal cells? So for this, we do mixes of DNA of 2 different cell lines. And several groups have done this, but I just did this experiment 2 months ago, and it looked so beautiful, I thought I'll show it to you. So once the DNA from one cell line has a segmental loss in chromosome 11. You see this small region right here, and it's a female. And the other cell line has a trisomy for chromosome 18, and it's a male. So I'm going to show you different ratios of mixes between these 2 cell lines. And what you would expect is to see a stepwise change as we move on and change the ratios. So initially, it's 100% this cell line A, and 0% cell line B. And I'll do 10% increments. So the second one will be 90 and 10, and then 80 and 20, and so on and so forth. So here we go. All right, let me go back on that. So you can look at the XY chromosomes, you can look at the X chromosome, and at the segmental, most importantly. And even that, so I'm going to narrate as we move along. So here it's the segmental is at 0%, 10%, 20, 30, 40, 50, 60, 70, 80, 90, and 100. So from this type of mixing experiments, we learned that PGT-A by next-gen sequencing can detect mosaicism at a very high accuracy. But then what should we do with those mosaic embryos? Maybe a few years ago, people would assume that they're abnormal, or actually we know that they have some abnormal cells in them. Should they be discarded? And this is just a summary of a presentation I gave yesterday, a study, a collaboration between many centers where we transferred 822 embryos that had the mosaic profile from PGT-A and compared them, the clinical outcomes, to the control group, which had 3,700 euploid embryos. So the implantation rate for the mosaic group was at 45.3% compared to 61% of the control group, the euploid embryos. And the ongoing pregnancy at time of analysis or birth rate was 36% for the mosaics and 52% for the control group. So even though mosaics have obviously a lower chance of positive clinical outcomes than euploids, many of them actually lead to pregnancies and births that are seemingly healthy. So far, we haven't heard of cases where these lead to compromise or abnormal births. Obviously, in a perfect world, we would just see the type of profiles I showed you, but some percentage of profiles we see in the lab look like this, where each lab then needs to decide what to do with this kind of embryos. We can get messiness or noise, and some of them fall within that range that you would define as mosaic. But the question is, are they true mosaics, or is this just an artifact? I will tell you, though, that if you re-sequence these samples, you will get 99% of the time you will get exactly the same result. So it's an issue of the cells, the input, basically. If you put in bad quality material, you will get bad data as well. In our lab, we treat these as like last priority, but we see that these can also lead to implantations and births. So in conclusion, so chromosomal uniformity in the trophectoderm biopsy is an excellent representative of the overall karyotype of the embryo. But sometimes we do detect mosaicism in the biopsy, but that can be done at very high accuracy with next-gen sequencing, PGT-A. Mosaicism in PGT-A should be its own category because embryos can implant, but they do so at lower implantation rates and positive outcomes than euploids. If you did not consider mosaics as their own category and you group them with aneuploids, you're discarding viable embryos. Or the opposite, if you group them with the euploids, then you're lowering overall likelihood of success. So I'll just conclude with this and want to thank these groups that collaborated in that big study of mosaic embryo transfers, and everybody at Zouves Fertility Center, and obviously our long-term collaborators. And I'll take any questions.